structure. They're all fine-tuned to be excited at slightly different wavelengths. Okay. Right, so this cartoon shows, again, just a diagram of what's happening in mitochondria versus what's happening in chloroplasts. And again, what we have is a situation where we've got a membrane. In the case of the mitochondria, it's the inner membrane. In the case of chloroplasts, it's the thylakoid membrane, so it's a lipid bilayer in either case. We have molecules that are involved in an electron transport chain in each case that are localized or anchored in the membrane. They generate a proton gradient across this membrane, and it's the diffusion of those protons back through ATP synthase that drives the production of ATP. So lots of nice sort of comparisons here, similar kind of overall pathway going on, but of course the actual molecules involved are really distinct from each other in each case. And this just shows a bigger picture of what's going on, where we have these photosystems and the associated molecules like plastoquinone and the cytochrome complex, plastocyanin, that are involved in these electron transport reactions, generating a proton gradient, and then it's those protons that flow back across the membrane through ATP synthase and drive the production of ATP. And of course, these molecules here, so ATP and the NADPH that's being produced at the end of this long electron transport chain, these are actually produced in the stroma, right? And that's important because that allows them to enter directly into the Calvin cycle, the enzymes that are involved in catalyzing the production of organic molecules and driven by this harvested energy. Right, so basically, in summary, the light reactions generate ATP. They increase the potential energy of electrons by moving them from water to ultimately to NADPH through this long sort of transfer process. So let's, why don't you pull out your iClickers and let's do one question. And I'm going to start the clock now. It'll take about 30 seconds or 40 seconds to do that. Okay, 5 seconds. 3, 2, 1. Okay, so 73% of you said that the answer is E. That's what I picked. I think the answer is E. Okay, and the reason is that, so a few people, so let's see, about 10% of you said the answer was B, that in addition to ATP, cyclic electron flow also produces oxygen and NADPH. So remember that if you look at the cyclic flow of electrons, that we're actually not making NADPH, right, in that cyclic flow. We're actually not doing that. We're generating ATP, but we're shunting it away from that terminal transfer pathway. Right? And then about 10% of you said that the answer was C. Only cyclic flow utilizes light at 700 nanometers. But actually, they both do, right? Because linear flow does as well, because in both cases, we're going through photosystem 1, which has that pair of chlorophylls that's excited at 700. Yeah. If an organism has both photosystems, it can do both, right? It can do this, it can transfer electrons to this linear flow, but it can also do the cyclic flow. Well, it'll tend to do more of the linear, right? Because it gets more bang for its buck doing that. Okay. Yeah. All right. So now let's talk about what is the consequence of all of this, right? So we've harvested some chemical, some energy in the form of chemical energy. We have ATP building up. We have NADPH molecules being produced. Those are both being released into the stroma. And so what happens then is that these molecules are actually able to help drive another cyclic process called the Calvin cycle, very analogous to the citric acid cycle that we talked about previously, in the sense that it has a series of enzymes that are catalyzing a cyclic set of reactions. But in this case, we're actually, instead of breaking down or oxidizing complex molecules, we're actually building them up, right? We're reducing, ultimately, CO2 into more complex molecules. So this is a process that, as we're going to see, it regenerates its starting material after molecules enter and leave the cycle, so that makes it a cycle. And it's building sugar molecules from smaller ones using ATP and the reducing power of NADPH to drive those reactions, which would otherwise be, or are, energetically unfavorable. We couple them to very favorable reactions, we actually can drive this process. And so in the Calvin cycle, carbon enters the cycle as carbon dioxide, and it leaves as a 3-carbon molecule called glyceraldehyde 3-phosphate. Now, you're right, you might remember that we've seen that molecule before. Do you remember where we saw it before? Glycolysis, right? Yeah, exactly. So it's not in glycolysis, and that's actually kind of important because you can see that if we produce glyceraldehyde 3-phosphate in the Calvin cycle, that actually is now an intermediate in glycolysis, right? So it can actually potentially feed right into that pathway in plants, and some of it does. So for the net synthesis of one molecule of glyceraldehyde 3-phosphate, the Calvin cycle has to take place 3 times because this is 3 carbons. We start with by fixing one carbon each time a carbon dioxide enters the cycle. So we have to fix a total of 3 of these to get one of these. And the Calvin cycle has 3 phases to it. The carbon fixation phase that's catalyzed by a really important enzyme called rubisco, then the reduction phase, and finally regeneration of the acceptor molecule for carbon dioxide, which is ribulose bisphosphate. Right, so let's just walk through the steps of the cycle. So we have carbon dioxide entering, and by the way, note that they're showing you this in terms of sort of thinking about a balanced equation here. Right? So we have 3 carbon dioxide molecules entering one at a time, and they react with this enzyme called rubisco that is responsible for this first step, which is called carbon fixation, in which ribulose bisphosphate, which is the acceptor, so the molecule that accepts the carbon dioxide, which is a 5-carbon molecule, is converted to a 6-carbon molecule, which is really very unstable. It's an intermediate that quickly breaks down to 2 molecules of 3-phosphoglycerate, which is shown here. Of course, again, keeping these balancing these equations in mind in terms of the numbers of molecules that are generated. Then, in the second phase of the Calvin cycle, this is called the reduction phase. And so what's happening here is that these 3-phosphoglycerate molecules are converted in a series of enzymatic reactions to glyceraldehyde 3-phosphate down here. And in the process, we actually have to hydrolyze ATP, and we also oxidize NADPH. So this is a very intensive, energy-requiring set of reactions here that convert 3-phosphoglycerate to glyceraldehyde 3-phosphate. And at this point in the cycle, we can actually this is where some output occurs. We can actually siphon off some of the glyceraldehyde 3-phosphate that's made here and use it for either building glucose, larger sugar molecules, or other organic compounds. It can also be broken down again, right? It can feed back into glycolysis potentially because it's an intermediate in that pathway. And then finally, over here, we have phase 3, the regeneration of the carbon dioxide acceptor, which is ribulose bisphosphate. And this is, again, a series of enzymatically catalyzed reactions that have to build a more complex molecule from these guys. And this is, of course, again, an energy-requiring set of reactions. So we, again, see ATP hydrolysis occurring here to make ribulose bisphosphate. And once we have this, we're back to the beginning. And this can then enter back into the cycle through reaction with Rubisco and fix carbon dioxide, go around the cycle this way. So let's do one more iClicker question at this point. And I'm going to start the timer right now and take about 30, 40 seconds for this. 30. It's pretty easy, I think. Okay, so about 5 seconds to go. And stop. Yeah, so it's good. So about 80% of you said that the answer is D, removal of electrons from chlorophyll molecules. Good job, that's right. Okay. Two people said A, creation of a pH gradient by pumping protons across the thylakoid membrane. And that's a consequence of light energy interacting with the system, but it's really not as direct as the excitation of chlorophyll, right? And then about 10% of you said that the answer was C, reduction of NADP plus molecules. And again, that's a consequence of that electron transport chain that happens that's triggered by light energy, but it's not the most direct thing that happens, right? The most direct thing that happens, the first thing that happens is the excitation of chlorophyll. Okay, so in the last few minutes, what I want to do is to just talk a little bit about some of the alternative pathways that have evolved in photosynthetic organisms to deal with particular kinds of environmental conditions. And so you remember that we talked about how carbon dioxide actually gets into these plant cells in the first place, these mesophyll cells where photosynthesis is going on, and they get in through these little pores called stomata, and oxygen can also get out of the stomata, and also there can be evaporation of water molecules through those pores as well. And so that means that when plants are growing in a very hot and dry climate, then they're really subject to dehydration because of this issue of wanting to allow access to cells want to get access to carbon dioxide, but by doing so in a hot, dry environment, they're actually losing water molecules through evaporation through those same pores. So to deal with that, plants will tend to close their stomata in hot, dry conditions or on hot, dry days, and this results in conservation of water molecules, but also tends to limit photosynthesis. And that's because the closing of these pores reduces the access to carbon dioxide, and it also tends to trap oxygen in those cells. And so the oxygen waste product can't get out, with oxygen concentration going up, carbon dioxide concentration going down. And when that happens, that tends to trigger what looks like kind of a wasteful process, and it's called photorespiration. And in photorespiration, which your book kind of speculates may be an evolutionary relic, and that's a possibility left over from conditions on the Earth when oxygen concentrations were really quite low and carbon dioxide concentrations were relatively very high. And fundamentally, this is a process that results from the fact that this enzyme called Rubisco, in addition to being able to bind and react with carbon dioxide, it actually also can bind and react with oxygen in place of carbon dioxide in the Calvin cycle. And so in photorespiration, this is exactly what tends to happen, because if we have a situation where we've got lower concentrations of carbon dioxide, we have higher concentrations of oxygen, then just by sort of relative concentrations, Rubisco will tend to react with oxygen instead of with carbon dioxide. And when it does that, it basically leads to the consumption of oxygen and organic fuel and the release of carbon dioxide without producing anything that's really useful to the plant, like ATP or more complex organic molecules, like sugar molecules. And so it's sort of a wasteful process. Question? Yeah, the organic fuel would be sugar molecules, right? So you're consuming them, right, in the process, and you're not building up any more. It's like the reverse process, right? So, right. And so, again, the idea is that maybe this is an evolutionary relic because Rubisco, this enzyme, first evolved under conditions on the Earth when the atmosphere was much lower in oxygen concentration and much higher relatively in carbon dioxide concentration. So it could be the case that it just didn't have to, there was no evolutionary pressure for this enzyme to be very selective for carbon dioxide binding and to exclude oxygen. Okay, but now that the sort of environmental conditions are a bit different, under certain situations, this becomes potentially an issue. But it also could be kind of helpful because it's thought that photorespiration can limit the damaging products of light reactions that build up in the absence of the Calvin cycle. And so when people have made mutations, made mutants in photosynthetic organisms that that are not as efficient at doing photorespiration, then they actually find that these plants are not able to grow very well under very intense light conditions. So it could be that there's actually a benefit to having this cycle as well, even though it seems to us, looking at it, that it would be sort of wasteful. Yeah. Well, because I don't know if it's necessarily favorable, but it's just a sort of one of the, if you just think about an enzyme that has the ability to bind to either of these substrates, if the concentration of one is a lot higher, it's going to tend to react more with that. And if that substrate is oxygen, then it's going to drive this pathway rather than the actual production process that leads to. How would this have evolved in conditions of higher carbon dioxide? Was more abundant. Well, I think what I'm really just saying is that, and this is just a theory, right, is that it could be that there was really no selective pressure for Rubisco to be more selective for carbon dioxide than it is. Because if it was evolving under conditions where it really didn't have to be, because there was so much carbon dioxide around relative to oxygen that most of the time it was interacting with carbon dioxide, there would be no selective advantage for that to evolve. Okay, it's a theory. It's the idea. So plants have evolved a couple of ways to deal with this, and I just want to mention these briefly. So most plants are called C3 plants, and that's because when they go through the Calvin cycle, that first product that's made is a 3-carbon molecule. And what happens in plants that are called C4 plants is that they can minimize the cost of photorespiration by first incorporating carbon dioxide into a 4-carbon compound. And they do this in mesophyll cells. And this requires a special enzyme called phosphoenol pyruvate carboxylase. You don't have to remember that, PEP carboxylase. And this is an enzyme that actually does have a much higher affinity for carbon dioxide than Rubisco does. So it can actually fix carbon dioxide into these 4-carbon molecules even when the CO2 concentration is pretty low. And so this requires a special structure in the leaves of these kinds of plants, because what happens is that these molecules are first made in the mesophyll cells, but they are then, these compounds are then exported into separate cells that are called bundle sheath cells, where they release carbon dioxide that is then used in the Calvin cycle. So it's effectively effectively a way of pumping CO2 into these bundle sheath cells in a way that, helps to deal with this problem of Rubisco having a relatively low affinity for CO2. And this is a cartoon that shows kind of how this works. So we have the C4 leaf anatomy has these mesophyll cells that surround bundle sheath cell, shown in the center. And in terms of the chemistry that's going on, we have carbon dioxide entering the mesophyll cell where it goes into this little cycle here where the first enzyme is PEP carboxylase. So CO2 is being fixed into a 4-carbon compound. And then it's able to, through enzymatic reactions, leads to the cleavage of this 4-carbon molecule to yield pyruvate, which can go back into this cycle. And with the release of carbon dioxide, and that step actually occurs in the bundle sheath cell. So the carbon dioxide can then enter the Calvin cycle as usual with Rubisco, but it's a very efficient way of segregating out the initial fixation of carbon dioxide over here and then introducing it in a way where it can be utilized by Rubisco over here. And then finally, there are plants that are succulents, for example, that are able to deal with this problem not by actually segregating these reactions in different types of cells, but they actually carry out reactions at under two different times of the light cycle, during the dark, at night, and during the day. And see, these are plants, again, many of them are succulents that fix carbon. These are chrysolation acid metabolism, named for the type of plants where this was first discovered, or CAM plants. And what they do is they open stomata at night, incorporate carbon dioxide into organic acids, and then close the stomata during the day and release CO2 from these organic acids into the Calvin cycle. And this last slide just shows a comparison of these two pathways. Here's what we just talked about with C4 plants, where these processes are physically separated into different cell types. Over here, this fixation is going on at night with release of carbon dioxide during the day to drive the Calvin cycle. So see you tonight if you're coming to Q&A at Dwinelle Hall, 7 o'clock.